Good morning, dear students of class 11 science. Welcome to biology class. We are with the chapter biological classification, chapter number 2, and we are dealing with the kingdom Monera. And under Monera, we are learning about bacteria. In the last class, we have learned about the classification of bacteria according to their mode of respiration, according to mode of nutrition, and so on. Clear? In today's class, we'll learn about the types of reproduction in bacteria. We'll learn about binary fission, conjugation, transduction, and transformation. Clear? So under the types of reproductions, we have to learn about the processes known as binary fission, conjugation, transduction, and transformations. There is no need to go in detail about the processes that occur during this type of reproductions. We only have to go through the proper definition of this process. Okay, so these are the four modes of reproduction which occurs in case of bacteria. So we'll go through it one by one. Then we will discuss about the differences between gram positive and gram negative bacteria. And before going with the differences, we'll first try to learn the structure of a bacterial cell. Okay, then we'll go with the difference. And lastly, we'll learn the economic importance of bacteria. Then we will finish the kingdom Monera. Then in the next class, we'll go with the next kingdom. Okay, so let us begin with the types of reproduction in bacteria. Okay, so generally when we talk about the types of reproduction in bacteria, it occurs by three processes. Okay, we say there is binary fission, and we have got sporulation, and we have got conjugation. But simply we will discuss about binary fission and we will discuss about conjugation. The sporulation is not there in your syllabus. Okay, so we'll first begin with binary fission. So in binary fission, fission means to break up. A single cell will split into two daughter cells. Clear? So under favorable conditions, binary fission is the most common method of multiplication or reproduction in bacteria. Now you know what is reproduction, right? Simply to reproduce or to give rise to the progeny. And we have got two types of reproduction, sexual and asexuals. Okay, so I'm not going into details about the difference between sexual and asexual. So mainly in case of bacteria, under favorable conditions, they used to multiply by means of binary fissions. Clear? So mature bacterium divides into two identical daughter cells by means of a mitosis. Okay, so during this process, the bacterial chromosome called the nucleate. Now what do you mean by nucleoid? If you remember that in your last class, I have shown you the structure or the genetic material of bacterial cells. Clear? In that class I have discussed you that the bacteria does not have a well-defined nucleus. That means the nucleus is not surrounded by a nuclear membrane. So therefore the chromosomal materials will be scattered throughout the cytoplasm in a structure known as nucleoid. Okay, but in case of eukaryotes, we have got well-defined nucleus so that the chromosomal materials will be packed inside the nucleus in a structure known as nucleosome, okay, along with the basic histone proteins that you have done in class 10. Clear? You have the word called nucleosome also, or heard the word called histone also. Clear? But in case of bacteria, they don't have a well defined nucleus. Clear? So in that case, the genetic materials will be present in a structure known as nucleate. So therefore during this binary fission process in bacteria, the bacterial chromosome, that is nucleoid, replicates to produce two nucleoids while attached to the mesosome. So look at the figure carefully now. Okay, so over here you can see the structure. This one is the nucleoid. Can you see over here? Right. And this one is the mesosome. Okay, inclusion in bacterial cells. So first of all, this particular nucleoid will divide into two nucleoids. Can you see here? This is the first nucleoid and this is the second nucleoid, or we can say daughter nucleoid. Okay, so the parental nucleoid has been divided into two nucleus. So this one is the daughter nucleate. Clear? But still, these nucleates are attached to the mesoderm. Clear? So whenever these parental nucleates will divide to produce the daughter nucleate, similarly, the mesoderm, alright, the mesoderm will also divide to produce the daughter mesoderm. So over here you can see the formation of daughter mesoderm and the daughter nucleoid. So therefore the nucleates are still attached to the mesoderm. Clear? Then after the formation of daughter nucleate, daughter mesoderm, there will be formation of membrane. Alright, new cross wall. There will be formation of membrane. So can you see over here? So a membrane formation occurs between the two mesoderm. Clear? And this membrane formation keeps on growing till it will divide the cytoplasm. Can you see over here? Till it will divide the cytoplasm and it will produce the two daughter cells. Okay, so membrane formation occurs, and this membrane formation occurs till it will divide the two cytoplasm and lead to the formation of two daughter cells. So from one parental cell there is formation of how many daughter cell? There is formation of two daughter cells. Clear? And these daughter cells will grow in size and they will repeat the process of binary fission. It's very easy. So what is binary fission? Mainly, the bacterial cell will divide into two identical daughter cells by means of mitosis. And first of all, in this process, the nucleate will divide to produce the daughter nucleate. Then the mesoderm will also divide to give the daughter mesoderms, so that the nucleus will be attached with the respective mesoderm. After that, the membrane synthesis occurs, or the membrane formation takes place between the two mesoderms. It will divide the cytoplasms and it will produce the two daughter cells. That means two daughter bacterial cells, and these two daughter bacterial cells will grow in size, and again they will repeat the process of binary fission. Okay, so the first mode of reproduction in case of bacteria, that is binary fission. Now let's talk about conjugation. Okay, so it is a primitive form of sexual reproduction which was discovered by Lederberg, Lederberg. Okay, it is a primitive form of sexual reproduction which was discovered by Lederberg, the name of the scientist. Okay, Lederberg, Lederberg, in E. coli. Clear? Now, what is conjugation? So it is mainly the transfer of genetic material between the two conjugates on coming in contact with one another. So we'll go in details afterwards. We'll go to the figure and it will be easier for you. Okay, but remember the definition. It is the transfer of genetic material between the two conjugates which are coming in contact with one another. Clear? And those bacteria that exhibit conjugations are dimorphic. That means they have got two sexual form, that is male and female. Okay, so we have got the male F plus or donor and the female F minus or recipient. So kindly remember this one. Okay, so those bacteria that exhibit conjugations are dimorphic. Di means two, morphic means form. There are two form, male form and female form. Okay, so male also called F plus or donor, and the female known as F minus or recipient. So afterwards we will talk why they are known as F plus, why they are known as F minus. Clear? So the male cells also possess pili, pili, and they have got F factor or fertility factor. So due to the presence of fertility factor, they are also known as F plus. Clear? And what is plus means? They are the extra chromosomal circular DNA molecule present in bacterial cells. Okay, this we have discussed already in our previous classes. But the female cells F minus, they don't have any type of such structures. They don't have pili or they don't have fertility factor. That's why they are known as F minus. Okay, and the transfer of genetic material between the two conjugates is completed by the process of sexduction or by the process of episome formation. Okay, so what is conjugation? It is the transfer of genetic material between the two conjugates on coming in contact with one another. And over here, kindly remember Thus, those bacteria which exhibits conjugations are dimorphic. Di means two, morpho means form. So there are two forms, male and female. Okay, male is also known as F plus or donor, and female F minus or recipient. That means over here the male is donating the genetic material to the female. Okay, so that's why male is known as donor and female is known as recipient. All right, so male possesses the fertility factor or F factor and pili, whereas such type of structure are absent in case of the female. Okay, and the transfer of genetic material, it occurs by means of sexduction or by means of episome formation. Now we'll go with the structures, that it will be easier for you. Okay, so first of all we'll just see the process of sexduction. Okay, so what happens in case of sexduction? Now first of all you have to see which one is male cell and which one is female cell. So can you see over here the F plus, this is the male cell, the donor, and this is the recipient one, the female F minus. Okay, so this is the plasmid, you can see over here, this is the plasmid and this is the nucleoid or the bacterial chromosome. And from the F plus, the transfer of genetic material must occur to the recipient. Clear? So the donor cell means these male cells get attached to the recipient cell. So the F plus cells will be attached to the F minus cells by the help of pili, P-I-L-I, pili or pilus. Can you show me a pilus? So by the help of this pilus or the pili, P-I-L-I, pili, the donor cells will be attached to the recipient cells. Okay, so in the region of contact, a pilus grow in size and it will form a cytoplasmic bridge. So this is the region of contact. Can you see over here? This is the region of contact where the donor cell and the recipient cells are coming in contact with one another. Okay, so in this region of contact, the pilus will grow in size and form a cytoplasmic bridge, or we also call it conjugation tube. Okay, now the plasmids of donor cell with the fertility factor F plus undergoes replication, means it will produce its carbon copy. Simple term. Okay, that means when the cytoplasmic bridge will be formed, then this F factor or fertility factor will undergo replication so that it will form its carbon copy. Can you see over here replicated F plasmids? So this F factor has undergone replication and has produced its own carbon copy or the replicated F plasmids. Alright. At this stage, a conjugated tube is formed through which the copy of fertility factor is transferred to the recipient cell. So when this fertility factor will be replicated, it will be transferred to the recipient cells by the help of this cytoplasmic bridge. And therefore, what happens is that now since the F factor has been transferred to the recipient cells, now finally you can see over here both the donor cells and the female cells now become F plus. Why we call it F plus? Female cell is basically F minus. But why are we calling F plus? Because now the female cell has received the fertility factor by the process of sexduction. So now both the male cell and the female cell, they have become F plus. Okay, so what is sexduction? The two cells has to come in contact with one another. What two cells? F plus and F minus. Okay, so these two cells will come in contact with one another by the help of pilus or pili, and in the region of contact between the two cells, pilus will grow in size and will form the conjugation tube or the cytoplasmic bridge. After that, the fertility factor of the male cells will undergo replication. It will produce the replicated F plasmids. Clear? And that replicated F plasmids will be transferred to the female cells by the help of cytoplasmic bridge. And this process is known as sexduction. After the transfer of fertility factor, the recipient cell, that is female cell, will also become F plus cells. Clear? Now let's come into the episome formation. Okay, the second mode of transfer of genetic material. Now, what is that? Sometimes the plasmid of the male bacterial cells will be attached to its chromosomes. Can you see over here? Look at this figure. So this one is the plasmid, and this one is the bacterial nucleoid. But over here, what I mean is that the plasmid is being attached to the bacterial chromosome. Okay, and whenever the plasmid will get attached to the bacterial chromosome, then such structure is known as epigenome. Clear? Okay, now such a male cell with epigenome is known as HFR cell. Now, what do you mean by HFR? Kindly remember, HFR means high frequency of recombination. Okay, so one more time, I'm telling you that sometimes the plasmids of the male's bacterial cell, that's F plus, will be attached to the bacterial nucleoid or the bacterial chromosomes, and such structure is known as epigenome. And such a male cell with epigenome is known as HFR. What is HFR? High frequency of recombination. Clear this one? Now, the episomes will cause a break in the bacterial chromosome so that some of the gene of the male cells are transferred to the female cells along with the plasmids. What I mean is that now there is formation of HFR. Now this HFR will lead to the formation of a break in the bacterial chromosomes. Okay, so you can see the formation of cytoplasmic breach or the conjugation tube. Now what I mean is that after that, not only the F factor or the plasmids will be transferred to the female cells, along with the plasmid, certain amount of gene will also be transferred to the female cells along with the plasmid. So the transferred gene recombine with the corresponding gene in the female cells. That means in that case, not only the plasmid is being transferred from the donor cell to the recipient cell, but along with the plasmid, the gene is also being transferred to the female cells or to the recipient cells. So now that transferred genes will become the part of the female cells. Clear? So these are the two processes where there will be transfer of genetic material in case of conjugation, that is extraction and episome formation. Okay, now next one, transduction. Now what is transduction? Transduction is the process of transfer of genetic material of one bacterium to another bacterium by temperate bacteriophage. Okay, now what is bacteriophage? Bacteriophage is a virus that will infect the bacteria. Okay, so in transduction, there will be transfer of genetic material. All right, so if there is transfer of genetic material, we require a portion of host DNA. We require a host. So from the body of the host, the genetic material will be transferred to the recipient one. So from one bacterium to the another bacterium, there will be transfer of genetic material, and for that one, we will require a bacteriophage, the virus that will infect the bacteria. And this process was discovered by Ginder and Lederberg in the year 1952 in a bacteria Salmonella typhimurium. Clear? So simple. What is transduction? It is transfer of genetic material from one bacterium to the another bacterium. And for this transfer, what do we require? We require a vector. Okay, a way, and that particular vector, that particular way or carrier is simply the bacteriophage. And what is bacteriophage? It is a virus that will infect the bacteria, and this process was discovered by Ginder and Lederberg in the year 1952 in a bacteria called Salmonella typhimurium. Okay, next one, what is transformation? Okay, it is the process of picking of DNA by a bacterium from the dead decaying bacterium. Okay, transformation is the picking of DNA by a bacterium from dead and decaying bacterium. And this particular process was discovered by Griffith in Diplococcus pneumoniae, the pneumonia causing bacteria. Okay, so this particular process you will learn in detail in class 12. This experiment. Okay, so what this Griffith observed? Griffith observed that the bacteria of non-virulent strain change to virulent strain. You learn in detail afterwards. That means non-virulent strain means it is not able to kill the host, and virulent strain means it is able to kill the host. But over here, what Griffith observed was that the non-virulent strain was able to change into the virulent strain, which when it was injected into the mice along with the heat-killed lysogenic bacteria of virulent strain. Okay, so this we'll learn in detail in class 12, but for now, kindly remember, Griffith first injected virulent strain into the mice. Which strain? Virulent strain. So when he injected the virulent strain into the body of mice, the mice was dead. Clear? And when he injected non-virulent strain into the body of mice, mice survived. Clear? Now what did he did? Griffith, by applying the heat, he killed the virulent strain. That's what we are saying. Heat killed virulent strain. So by applying heat, he killed the virulent strain. So when he injected the heat killed virulent strain into the body of mice, the mice did not die. When he injected heat killed virulent strain into the body of mice, the mice survived. It did not die. Clear? Now, in next step, he put the mixture. Listen very carefully. Now, he put the mixture of heat killed virulent strain and non-virulent strain. Which mixture? A mixture of heat killed virulent strain plus non-virulent strain. Now, when he injected both the strain separately, the mice did not die. But when he injected the mixture of these two strain, non-virulent strain and the heat-killed virulent strain, the mice was dead. How? How was it possible? You observe that in this case, that heat-killed virulent strain was able to transfer the non-virulent strain into virulent form so that the mice died. So in that case, okay, so in this case, the genetic material, that is DNA, was picked up by non-virulent strain from heat-killed virulent strain. Okay, so this is the process of transformation where the non-virulent strain was changed into virulent strain. Clear? So, Avery et al. In the year 1944 found that the virulent strains are developed due to the transfer of DNA segment from the dead cells of virulent strains to the living cells of the non-virulent strain. So due to the transfer of genetic material from the heat-killed virulent strain into the non-virulent strain, the non-virulent strain was changed into the virulent strain, and this process is known as transformation. Clear? So we have finished the types of reproduction in case of bacteria. Now let's go back into the structures of our typical bacterial cells. Okay, so before going into the differences between the gram-positive and the gram-negative bacteria, we'll first try to look at the structure of a bacterial cells. All right, so bacterial cell is composed of a rigid cell wall, which thickness is about 7.5 nanometer to 12 nanometer in gram-negative bacteria and 20 nanometer to 80 nanometer in gram-positive bacteria. So for this, kindly go through the page number 10. Okay, 2.10. 2 means chapter number 2 and 10 is page number for the chapter number 2. Okay, now this cell wall, it just lies outside the plasma membrane. And cell wall is mainly formed of peptidoglycans, mainly a polymer of sugar and amino acids that we have learned already. Okay, then we have got few lipids, we have got phosphorus, inorganic salts, and the derivatives of glucose called muramic acids. These are the composition of cell wall in case of bacteria. Now, gram-negative bacteria also had another covering. Generally, the cell wall is made up of peptidoglycans, but gram-negative bacteria has another covering also called lipopolysaccharides. Along with peptidoglycans, gram-negative bacteria, they have another covering called lipopolysaccharides. So kindly remember this term. Lipopolysaccharides are present in case of gram-negative bacteria. Okay, though they are made up of peptidoglycans. In both gram-positive and gram-negative bacteria, we are finding the cell wall made up of peptidoglycans. But in case of gram-negative bacteria, along with peptidoglycans, we will find another covering called lipopolysaccharides. Clear? Now, we'll see, sorry, okay, we'll just talk about instance of gram-positive and gram-negative bacteria. Okay, kindly go to the page number 10 only. Over there, you will find a Danish bacteriologist called Hans Christian Gram. So kindly remember this term name, okay? A bacteriologist called Hans Christian Gram, who stained bacteria. Stain means to impart color. Okay, who stained bacteria with crystal violet stain. Which stain? Crystal violet stain. Stabilized with 0.5% iodine solution and subsequently washed with acetone or alcohol. So he stained bacteria with crystal violet, stabilized with 0.5% iodine solution, and after that he washed with acetone or alcohol. Clear? He found that some of the bacteria they turned into deep violet or purple, but some bacteria did not retain the stain. So some of the bacteria they were able to retain the stain, they were able to absorb the stain, and those bacteria which was able to retain the stain, which was able to absorb the stain, they turn into deep violet or purple color, whereas some of the bacteria they were not able to retain the stain. Now when this bacteria, that means those bacteria which was not able to retain the stain, which did not turn purple or violet, so those bacteria when they were stained with safranin or carbofulcin, colorless bacteria become red. Okay, so those bacteria they turn into red color when they were stained with safranin or carbofulcin. Bacteria which remain purple are called gram-positive bacteria, and those bacteria which turn red are called gram-negative bacteria. So now we'll go to differences. Okay, gram-positive means they will retain the gram stain. Now what is gram stains? The staining which are done by Hans Christian Gram, where he stained the bacteria by using crystal violet stain stabilized with 0.5% iodine solution and washed with acetone or alcohol. Okay, it's called gram stain. So those bacteria which was able to retain the stain, they were called gram positive bacteria, and their color is to be purple or deep violet. And those bacteria which are not able to retain the gram stain, they were known as gram negative bacteria. Now gram positive bacteria, point number 2, is single layer, that it is composed of peptidoglycan. And go to gram negative, they are composed of two layer, that is cell wall is multi-layer, sorry, is two layer. And what are the two layer? First layer is peptidoglycan and the second layer is lipopolysaccharides. Clear? So due to the presence of the second layer lipopolysaccharides, gram negative bacteria were not able to retain the gram stain. Okay, point number 3, cell wall in case of gram positive bacteria is 20 nanometer to 80 nanometer, whereas in case of gram negative it is 7.5 nanometer to 12 nanometer. Gram positive bacteria they used to lack pili or fimbriae, whereas in case of gram negative both of them are present. Okay, in gram positive bacteria, point number 5, cell membrane invaginates and gets folded inside the cell forming the mesosome. Now which one is mesosome? If you look in the structures of the bacterial cells, there will be inclusions. Can see over the inclusions? The cell will be folded insides. So this inclusions is known as mesosome. Okay, so these mesosomes are present in case of gram positive, but they are absent in case of gram negative. Okay, gram positive cell wall contains traces of lipids, but in gram negative there are 20% of lipids. And in gram positive there are a few pathogenic forms which are used to cause disease in case of the host, but in gram negative there are more pathogenic forms. Example, in case of gram positive Streptococcus, Bacillus, Clostridium, Streptomyces. Okay, and grounding examples: Salmonella, Pseudomonas, Vibrio, Rhizobium, and Listeria. Clear? So the simple differences between gram-positive and the gram-negative bacteria. Clear? Okay. The next one, we'll talk about the economic importance of bacteria. So first over here, we'll discuss about the role of bacteria in the sewage treatment. I think you have heard about the sewage, which contains lots of pollutants, like we can say lots of microbes are there, right? Lots of we can say pollutants like paper, plastics, right? So lots of household pollutants or waste products are present in case of sewage. Okay, that means highly polluted one. And if we used to discharge this sewage directly into the water body will be polluted. And if the water body will be polluted, then it will lead to the death of aquatic plants and animals. So therefore, before discharging this sewage directly into the water body, we used to go for the treatment of sewage. And in sewage treatment, there are a few bacteria which are involved. Okay, so this we will learn in details in case of class 12. But just for now, remember that for sewage treatment, we use the bacteria called methanogens. Methanogens are those bacteria, all right? Methanogenic bacteria are those bacteria who used to produce the gas called methane. Okay, so there are two methanogenic bacteria, Methanococcus and Methanobacterium. Remember this name. Okay, Methanococcus and the Methanobacterium are the two methanogenic bacteria who are employed in case of sewage treatment, and they are mainly responsible for the formation of methane gas during the sewage treatment. Okay, so they will decompose anaerobically, means in the absence of oxygen, the solid organic waste of the sewage into methane and carbon dioxide. Okay, and mainly this mixture of gases, methane and carbon dioxide, is called biogas, which mainly used as biofuels. Clear? And then mainly this biogas will be used as a fuel, and the remaining one will be used as a sludge for the fertilizer. Okay, next one, most important, is antibiotic. So antibiotic is mainly a chemical that is produced by the microorganisms and is mainly used for the treatment of different types of fungal infections and bacterial infections. So man has isolated many of the antibiotics from various bacteria and have utilized them as drug for the treatment of different types of pathogenic microorganisms. And what was the first antibiotic? It was penicillin, discovered by Sir Alexander Fleming. Okay, obtained from which fungi? Penicillium notatum. Okay, so we'll go through the examples of few antibiotics, their sources, range of actions, and their mode of actions. It's in the page number 19. Okay, table number 2.7, page number 19. So first one is colistin, the name of the antibiotic, also called polymyxin E. Source is Bacillus colistinus. Range of actions: it is effective against gram-negative bacteria. How does it work? It will destroy the cell membrane. Okay, it will destroy the cell membrane of gram-negative bacteria and will kill the bacteria. Number two, tyrothricin, the name of the antibiotic. Source is Bacillus brevis. Range of actions against gram-negative bacteria. How does it work? It will inhibit the formation of bacterial cell membrane. Inhibit means to stop. Okay, it will stop the formation of cell membrane in case of bacteria. Number third antibiotic is polymyxin G, and over here the source is Bacillus polymyxa. The range of action is against gram-positive bacteria. It will work by destroying the cytoplasmic membrane. Okay, number four antibiotic is bacitracin. It is obtained from Bacillus subtilis, and range of action is against the gram-positive bacteria. It will work by inhibiting the synthesis of bacterial cell wall. Okay, so these are the name of few antibiotics, their source, range of actions, and the mode of action. So kindly remember the name of antibiotics along with the source, range of actions, and the mode of action. Now next one, bacterial role in case of energy productions. So for your first one, you have learned about the biogas productions or biogas formations, where we will use the methanogenic bacteria, Methanobacterium and Methanococcus. methanococcus. Okay, so kindly remember that name, methanobacterium and methanococcus. So they are mainly used in the formation of biogas. Then we have got another bacterium called Clostridium and E. coli. All right, from where we have developed the hydrogen gas. Okay, scientists have produced hydrogen gas from this particular bacterium, which is mainly a source of clean energy. And this particular energy can be used in motor vehicles and also to generate electricity. And last one is the role of bacteria in household products, where they have been utilized for the formation of curd and cheese. And for this one, the name of the bacterium which is employed is lactic acid bacteria called lactobacillus. So kindly remember the name of the bacteria, lactobacillus, which is also called lactic acid bacteria, is being used in the production of cotton cheese, the household products. So today we have simply discussed about the types of reproduction in case of bacteria, and we have discussed about the difference between gram-positive and gram-negative bacteria. We have seen the structure of bacterial cell wall, then we have gone through the economic importance of bacteria. Okay, so this much for today. See you in next class, and in the next class we'll begin with the kingdom protista. Thank you.